Thanks very much. It's a great pleasure to be here, and I want to thank the organizers for giving me an opportunity to tell you a little bit about the kinds of things that we're doing. And the last two talks we heard, I think, are a great introduction to what I want to talk about, which is really diversity. And so if we think about diversity and what we've learned from both of these talks really encompass diversity in terms of what that diversity represents and what we might be able to do with that once we mind it a little bit. And so, you know, if I had to make my $2 billion plug in terms of what we might want to think about, it's trying to figure out who are we sharing the planet with? What are those organisms, and what are they capable of doing? And I think we heard the last two talks a little bit about some of the discoveries that could be made. And so what I want to talk a little bit about is just how little that we know. So, so really, there's really three goals that I want to do today. First of all, you probably wouldn't have heard viruses very much, or most of you in the kind of context I want to talk about them. But I want to make the case that most of the biodiversity on the planet is actually found in viruses. Secondly, I want to convince you of the importance of viruses other than as purveyors of disease. So I want to really leave you with the idea that if it weren't for viruses on the planet would probably not exist. At least we wouldn't exist, life wouldn't exist on the planet. And third, that show how genomics is the lens through which we can unlock this diversity and hopefully potentially make some remarkable discoveries along the way. So this is a symposium about genomics, the power and the promise. And so I think the power, of course, is that genomics is allowing us to unlock this diversity. The new technologies in sequencing are really uncovering diversity which you never knew exist. And I think the promise is that this diversity has untold riches, basically, that we'll be able to use for all kinds of things and also just to understand how the ecosystems function. And we heard, again, a couple of nice stories about what that diversity represents. So one of the things that's important, I think, to think about is what is biodiversity? And I'm actually work in a biodiversity institute, and sometimes my colleagues will say, you don't really work on biodiversity, you work on genetic diversity. And so I think that I really like this definition, and I think it's one that my colleagues and myself can accept, is that really biodiversity is the totality of that genetic information, the species that are there, and the ecosystems in a region. And in fact, this was a definition that was agreed upon after a very large round of consultations and has ultimately been adopted by the United Nations as well. So if we come from that premise, basically what we're saying then is that we know that if we have no genetic diversity, we have no biodiversity, right? So we know that species richness ultimately stems from the underlying genetic diversity, and then ecosystem richness and diversity ultimately then underpins genetic diversity. And so if we can agree on that, if we can agree that ultimately biodiversity is genetic diversity, then we need to start thinking about what that genetic diversity is and what it represents. So the roots of biological diversity then and ecosystem function ultimately reside in the overall genetic diversity. So hopefully that is something that we can agree on. So if we think about marine biological diversity, it's really quite interesting because our understanding of the oceans has changed remarkably in the last few decades. And if we think about originally, it was just looked upon as a vehicle for transporting goods. We would go out there, we would mine it for resources, it was a source of fisheries, things like that. But now, I think, there's been this transformation. We're starting to look again at the oceans as this vast reservoir of potential genetic and biological diversity that we can go out and perhaps make inquiries, interrogate that diversity, and try and perhaps understand a little bit about what it's doing and its function. So most of life in the oceans is microbial, and many of you may not think about that, but if we think of the protists, the bacteria, and the archaea, and the viruses, in fact, that represents just about everything that's in the ocean. So in fact, if we took everything in the ocean that we can think about, so everything that maybe we would be familiar with, or that our family members might be familiar with, and you could put it on one side, so the fish, the whales, the algae, the crustacean, the zooplankton, and everything like that, you could put it on one side of a giant balance. And on the other side of that balance, you could put microbes. 95 to 98% of the living material by weight in the oceans is microscopic. You can't see it. Okay? So that 95, 98% of that material produces half the oxygen on the planet. Okay, so it's clearly incredibly important in terms of ecosystem. So again, if we think about biodiversity, ecosystem functioning, the balance of the planet, we need to think about the microbial life that's there. And so we can look at that by weight. So what is that microbial biomass? So if we look at it in kind of a big pie chart, most of it is prokaryotic. Most of it's bacterial. A significant amount of it is archaea. And then we've got the viruses and the protists make up roughly equal amounts of the remaining. And again, the rest of the big stuff, the whales and all those things we like to think, the charismatic megafauna, in fact, are actually pretty trivial in terms of overall biomass in the oceans. So we could look at it this way. So if we were to look at viruses, there's about 200 megatons of carbon in viruses in the ocean, which is equal to about 75 million blue whales. Okay? If we looked at whales, the most biomass that was ever in whales, as far as we know, is about 13 megatons. Okay, and that's completely dwarfed by the carbon that would be in the prokaryotes, which is about 5.2 gigatons. So again, I always tell my class, if you're interested, whales are great. They're very top predators. But if you're actually interested in how the planet functions, it's not the whales that we need to be concerned about. It's that microbial life that we can't see. If we look at by numbers, though, it's quite different. So by viruses, there's about 10 times as many viruses in the ocean as there are bacteria. And then protists, again, are about tenfold or more less than the amount of bacteria that are in the ocean. So by abundance, it's viruses. So in fact, in a liter of seawater, there are more viruses than there are people on the planet. Okay, so if we were to take a drop of seawater, this is pretty much what it would look like. So this is a drop of seawater, which is stained with a nucleic acid stain, in this case YoPro, but a lot of people would use Cyber now, Cyber Green or Cyber Gold. And all of these little dots, in fact, that you see there, those are individual virus particles. The ones that are slightly larger are bacteria, and that big yellow blob that you see, that's a protist. So if we were to come to Earth from another planet and kind of try and sample it, this is what we would most likely see. We would see a planet which is completely dominated by microbial life, most of which would be in viruses, the rest in bacteria and protists. Okay? So on average, there's about 10 million viruses and 1 million bacteria in every liter of seawater. And that's also true of freshwaters. Okay? It's in that same general range. So if we think about it, we compare that to stars in the universe, for example. Well, there's about 10 to the 23 stars in the universe, and there was a paper that came out a few years ago because they had underestimated the number of stars in the universe by threefold, right? So in biology, that probably wouldn't garnish you a paper in Nature, but if it's counting stars in the universe, it does, okay? In contrast, there's about 10 million-fold more viruses in the ocean than there are stars in the universe. And so we can think about that in sort of gigantic proportions. So one day I was, probably should have been writing a proposal for Genome Canada or something like that, but I was sitting at my desk and I thought, well, if we took all those viruses in the ocean, we stretched them end to end, how far would they go? So I really didn't have any idea, maybe the moon, maybe the sun. So I just, sat down and did a few calculations on the back of the envelope. It was easy to figure out how many viruses are in the ocean, about 10 to the 30th, because we know the volume of the ocean. And I just took the minimum abundance of viruses that you find anywhere in the ocean. That's about 1 million per mil. And then I figured about the average length of a virus is about 100 nanometers long. So if we take 100 nanometers times 10 to the 30th, we end up with 10 to the 23 meters, which is about 10 to the 20th kilometers, which is an extremely large number that I had trouble thinking about. I have trouble with the US deficit, never mind the size of this number, right? So if we take that and divide it by kilometers, we end up with 10 to the 13th, we end up with 10 to the 20th kilometers. You just divide that by 10 to the 13th kilometers by light years, we end up to 10 to the 7th light years. So if you stretched all those viruses end to end, they would go 10 to the 7th light years. So the nearest star is Proxima Centauri at about 4.2 light years away. The Crab supernova is 4,000 light years away. Our own galaxy, the Milky Way, is about 150,000 light years across. In fact, they would stretch further than the nearest 60 galaxies. And so that may seem like a trivial calculation, but I think it's important on a number of levels. For one reason is that what we have then is about Avogadro's number of infections per second in the ocean, about 10 to the 23rd, 10 to the 24th infections per second in the ocean. And so each one of those events is an opportunity for evolution to occur, for lateral transfer of genetic information. Each one of those virus particles has its own genome, right? So it has its own genetic information, its own genetic identity. And so if we start thinking about what that means in terms of where most of the genetic information on the Earth resides, it actually resides in viruses. So the number of viruses in the sea is enormous. So why does anybody care? Well, 95 to 98% of the biomass in the ocean is microbial. As I mentioned, the oceans produce about half the oxygen on the planet, and viruses kill about 20% of the living material in the ocean every day. So what we end up then with are viruses are incredibly important in driving global planetary cycles. And so before we would think on that top line there, you've got phytoplankton going to zooplankton going to fish, which is I don't think anybody for a long time has thought that's exactly the way ocean food webs work, but that's kind of the basis of what we would think about. And so what we now have incorporated into our thinking is something which is called the viral shunt, which essentially is moving that material from what we would call particulate living material into particulate dissolved in organic matter, and ultimately that cycles back through heterotrophic bacteria and produces CO2. So the pathways of nutrient and energy cycling on the planet have really changed as a result of our understanding what viruses are doing. And what's really interesting, if you take viruses out, the ocean photosynthesis stops in the ocean. So that was a big surprise. So it turns out that viruses are major drivers of nutrient cycling in oceans. Okay, so hopefully I've made the case at least that viruses are important other than just purveyors of disease in humans and animals and crop plants. Okay, so if we start thinking about diversity, so we've talked about that a little bit in the big tree of life. This is going to be pretty elementary for most of you here, but if we look at, this is a ribosomal RNA depiction of life's diversity, and up in the corner there we have bacteria, and then quite distant, but also very small, are the archaeans. And for the first 3 billion years or so of life on the planet, that's what it looked like. And then the protists came along, the eukaryotes. And so that's basically the diversity of life we see in the planet. And so some people might rightfully ask, well, where are the higher organisms? And I guess if I had to throw in a higher organism, I'd throw in like a mushroom or something, right? And so, but what about us? Well, there I am. And so you're probably slightly more closely related to me than a mushroom, at least. Perhaps some of my relatives are closer to mushrooms, but overall, that's where we heard about transcription and translation of genes and regulation. So with a little bit of that kind of DNA, I'd probably be a mushroom, right? So we carry most of the same genetic information, mushrooms and us. The rest of the tree, literally everything below that is microscopic. So if you look at the diversity of life on the planet, everything that's tied up in macroscopic organisms that we can see, the genetic diversity is actually pretty trivial, right? So most of the novelty, most of the things that, for instance, we heard about in the last talk, that's all encompassed in those things that are too small for us to see. All that genetic innovation, right, that took place over billions of years. Okay, so what about viruses? Well, they're so distant we can't even plug them into the tree of life. And the consensus is right now that's building is that viruses were there before cellular life. So ultimately, they came out of that same pool of genetic information that cellular life came out of, and we don't really understand how it was all put together. But you cannot build viruses from cellular genes. Okay, so what do? How do we catch viruses? Well, we can go on a plane. That's probably one of the ways that most of us catch viruses. But we do go out in ships and things like that to kind of remote and exotic locations. We collect large volumes of water, and we remove the viruses from that. And we actually have a library now of about 2,000 concentrated virus communities that we've collected all over the world. And so one of the first studies we did was a collaboration with Forrest Rohrer at San Diego State University. And so we just asked the question. We took some of these big communities of viruses that we had and asked, well, what's out there? So what kinds of things, what kind of genetic information is encompassed? So we took a metagenomic approach with the early next-gen sequencing technologies. So we took 56 communities from the Arctic Ocean, 85 viral communities from the Pacific Ocean, 44 virus communities from the Gulf of Mexico, and we took one from the Atlantic Ocean. And so we pooled this information together, and we looked at it in different ways. There's lots and lots of stories that one could tell about that. There's lots of interesting things that came out of it. But one of the most remarkable things was that about 90% of the coding sequences that we pulled out from the viruses had no homologs in any database that we could find, no obvious homologs. So that's remarkable, because if we think about 90% of the coding sequences not having any genetic similarity to what was known, remember that all of those things are coding for proteins with function, right? And so that means that we have absolutely no idea what these things are doing. And we still go out now. This was pretty short reads. This was early days. Now we take longer read libraries, for example. We're still at about 80%, even with all the metagenomic sequencing that we've done. We're still 70 to 80% of the coding sequences that we pull out of the ocean still don't have any homologs, even with other databases of metagenomic data from other environments, right? So the genetic diversity is enormous, and we're still a long ways from closing that sequence space. Okay, so that was DNA virus. We could look at RNA virus. This is Alex Cully in the top, who is a PhD student of mine. Andrew Lang, who was a postdoc, went out and said, well, what if we asked the same question about RNA viruses? And the gray is what you see in terms of the proportion of sequences, again, that had no homologs in any database, and it varied between 60 and 80%. And yet those samples were taken 100 kilometers apart from each other, and there was literally no overlap amongst the ones that were known, even in families of viruses that were represented. Okay, so again, the diversity out there is enormous and largely unknown. If we look at marine single-stranded DNA viruses, Jessica Labonte, a graduate student in my lab, was just finishing up. So we decided we would purify single-stranded DNA from the oceans, which is great because that's only viral DNA. And so what she found was much like what we found in other studies, is 90% of that single-stranded DNA had no homologs in any database, and then that 10% that did essentially encompassed all the known families of single-stranded DNA viruses. So 90% of it didn't correspond to any of the families of single-stranded DNA viruses that were known. And then through a series of sort of sophisticated network analysis and genomic analysis, we were able to actually close more than 600 viral genomes completely, and ended up with 125 distinct evolutionary groups that had no relationship to each other or any relationship to anything that was known. Okay, and so there's 7 or 8 families of recognized single-stranded DNA viruses. We have potentially another 118 new families of single-stranded DNA viruses pulled out of a few samples of water. Okay, so the diversity out there is enormous and really, really largely unknown. So why does anybody care? So again, most of the genetic diversity on Earth is microbial. Much of that diversity remains within viruses, and just about all of this diversity still is unknown in terms of what it is and what its function is. So the promise, can any of this information be translated into application? Well, that's a difficult problem, right? Is how do we take this genetic information and translate it into something which we might be able to have some understanding in terms of what its function is? Well, we can look at what's been happening in terms of marine genetic patents, and we can say, well, amongst the stuff that's known, right, so where we actually do have homologs to genes and we can recognize its function and we can capture that function, we can see that there are quite a lot of patents that have been come out with potential application, ranging from human health to biofuels to bioremediation to biotechnological tools. There's lots of things. And if we look at how that's changed, we can see that over many years, people were purifying natural products from the ocean, anti-cancer compounds, lots of things like that. They were looking at natural products. But now if you look at sequences that are being patented because of putative function, that they might have some benefit, you can see that that's been increasing incredibly rapidly. If we look at who's doing that patenting, it reveals another kind of problem. And so we can look at these data and we can see that the patents are essentially entirely encompassed by those countries which are certainly the most developed, right? And certainly have the most potential for going out and being able to benefit from this genetic information. So you can see on that little embedded graph there, which is a log log plot, you can see that on the x-axis is the number of patent claims, and you can see that most of the patents are held by less than 1% of the countries. And so then this brings up other questions that we need to at least be thinking about, and that is who owns these genetic resources, right? So are these genetic resources just there for exploitation by the most developed countries on the planet, or in fact, is this a global resource that we somehow have to figure out how to be able to share in? And that becomes complicated again, so the United Nations and UNCLOS is trying to work on these questions, because microbes, they just kind of move around, right? So you can't really say they don't stay in territorial waters, for example. And so I want to tell you one other little vignette, and it's not just about the diversity that we find from genomics and metagenomic analysis. You can actually go out there and you can actually grab viruses and you can isolate them and you can kind of characterize them biologically and genetically. And so this is a story about one giant virus that one of my graduate students worked on. It infects an organism called cafeteria. Cafeteria is a little phagotrophic zooplankton which eats bacteria for the most part. And we had isolated a large virus, which actually, that zooplankton, which actually kills that zooplankton. And it's one of the largest viruses that was known. It's 730,000 base pairs of coding sequence. So, and if you look at that again, it's the usual thing, although in this case, there was more homologs than we would typically find, but you can see that that genetic information comes from eukaryotes, bacteria, archaea, other large viruses, and other kinds of viruses. So in fact, I think one of the things that's really cool is I believe, I have to be disproved, that viruses essentially archive all the genetic information on Earth, are archived in viral genomes. Okay, so if we wanted to look at where it is, we could go out there, and I think if we pulled out all the viruses, we would find homologs of every gene that's known, but interestingly, most of the genes in viruses have no homologs in cellular organisms. But viruses contain homologs of most genes that are found in cells. In fact, there's viruses out there that encode the genes for photosynthesis. This virus is one of the most complex viruses known. For example, it has more than 40 proteins associated with DNA replication. It has most transcription programs, translational programs. It has 9 proteins involved with DNA repair, and it can in fact repair its own DNA outside of the host through photoreactivation, for example. So highly sophisticated. But one of the neat things, and this is where the story is that I want to kind of tell you about, is that when we looked at these viruses, here's big viruses, they replicate in something called the virus factory. We saw all these little viruses there as well. And so these little viruses, which are shown in red, are obligate parasites of the big virus. And so the zooplankton, if it's infected by the big virus, actually goes out and transports in the little virus, which kills the big virus and rescues the cell. So it's actually an adaptive immune system. You could think about it, about pulling in a virus which kills the big virus. And so then we asked, well, what is this little virus? And when we looked at this little virus, here you can see the little virus and the big virus beside each other. When we looked at the little virus, which we call mavirus, is in fact not related to other viruses at all. It's related to large transposable elements that are found in higher eukaryotes. And so the hypothesis here is that over evolutionary time, that cells that were able to incorporate this genetic information into their own genomes were able to resist infection by these large viruses, and in fact were immune to them, so that these large viruses ultimately died off. And so what we have now are all these transposable elements, which have got all kinds of variation in them, jumping around genomes, but they really don't have any function anymore because there are no large viruses like this which are infecting higher eukaryotes. They're extinct. So there's a nice little story. So I think the bottom line here is to kind of wrap up is that The Economist kind of picked up on this story, and the idea is that biodiversity is really a lot more complicated than we think about it. And this came out while the Nagoya Conference on Biodiversity was going on, and you know, it's eloquently stated here that it's not just a matter, diversity is not a matter of tigers and whales or butterflies, for example, but it's about all those little things, all those microbes, which are too small to see and are too numerous to count and do things in so many ways that we can only imagine. And the last thing I want to do are what about this biodiversity? Are we in danger of losing it? So I think it's a while before I'm going to be able to convince Greenpeace. I saw a Greenpeace slide earlier, but I haven't seen them go up there waving "Save the Virus" yet, right? So I've got a little bit of ways to go before I can get there. But one of my graduate students works on the Markham Ice Shelf, which is on northern tip of Ellesmere Island. That's where people, when they want to go to the North Pole, they go there, and then they take off across the ice and hope they get there before they die, right? So, but the Markham Ice Shelf, that's an ice shelf that's been there for a long, long time. And one of the things that's really interesting about these ice shelves is there's these long fjords or these inlets behind them. And these ice shelves dam the front of those fjords, right? They form a giant ice dam, many, many meters deep. And so it isolates the water behind it from the ocean water. And we know from that initial work that we did with Forest Rowers Group that, for instance, the viruses, the genetic information of viruses in the Arctic Ocean is completely different than the genetic information of viruses in the Pacific Ocean. There's all kinds of genes that there's just no overlap in at all. And we also know in these giant ice dam lakes that have been there for 25 or 30,000 years, the genomic information, the genetic diversity in there is completely different than what we find in the Arctic Ocean. And so in a period of a few days in 2008, the Markham Ice Shelf broke off and the ocean just flooded in to that fjord, and that ecosystem was completely gone in a matter of days, without us ever really knowing what was there. And so I think that's something that we need to think about because again, unless we know what we're losing, unless we know what's there, we just have no idea of what that potential is, right? And so I think that there really is a case to be made that this isn't something that's going to be translated into a product in 5 years. But ultimately, it's that genetic information out there which is going to be the source of new ideas and new technology that ultimately could be of enormous benefit. So the take-home message is there. Hopefully I've convinced you, if you weren't, that genetic diversity underpins biological diversity, that the marine microbes encompass most of the genetic diversity, unknown genetic diversity on Earth, and that it's ultimately the identification, development, and preservation of these resources and their biological potential that will ultimately be really important down the road. And finally, I want to thank all the people that make it possible. These are extant people in my lab, but all the ones that have been there in the past, and also all those agencies that allow us to go out there and just explore our ideas. Thanks very much.